Hi, friends. So, I want to talk about truth, justice, the American way, and a bunch of other stuff, but specifically about a basic claim that is true. Namely, that it's likely coronavirus is a bioweapon that was possibly released accidentally, probably, or maybe purposely, or whatever. This is the most convincing article I've ever read regarding the matter, and there's a good reason why my NI is now telling me, okay, we can draw some pretty clear conclusions here. Here's the key thing. This Yan person who's made the claim that it's man-made has been subject to a lot of rebuttal. Shoddy Bannon-backed paper. In fact, not man-made or engineered, but its origins unclear. Fact check. Um, Here you see that. Oh, not in this one, I guess, but there's a Fox News thing that mentioned this, and now it says not true information. 8 persistent COVID-19 myths and why people believe them. That it was engineered in a laboratory in China. Why it's false. Why people believe it. People want a scapegoat. Blah, 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 blah. So, you'll note here why people believe it. People want a scapegoat for the immense suffering. Accidental pathogens sometimes do occur, accidental releases, and it provides enough legitimacy to demonize China. Okay, so but here's why people actually believe that it might be man-made. Likely is. Notice it says why people believe it. This is bullshit, right? This is not why people believe it. The reason people believe it is because of these arguments. If we want to engineer a virus, we manipulate the biological sequence. A sequence means the order in which the basic biological letters occur, and this order determines the function and purpose of the sequence, just as the meaning of words is based on letters. So, as published by Yan, a search of GenBank reveals that the viral envelope protein found in the novel coronavirus is 100% identical to two other strains in the database, ZC45 and ZXC21. These strains are the ones Yan mentions as the foundation for the new strain and were submitted to GenBank more than 5 years ago. The gene looks like it was copied from one of the two coronavirus strains and placed into SARS-CoV-2. 100% identity is rare across animal species and is nearly unknown across virus species and rarely occurs among different viral strains. One possible explanation is that a rare recombination event occurred naturally. It's hard to imagine that a viral gene replicating in bats collected more than 5 years previously would retain protein identity and all but 3 nucleotides in the wild, but the possibility should be entertained. The other anomalies in the viral genome Yan mentions have explanations with varying degrees of plausibility as to why they might have occurred naturally. It's impossible to prove that something occurred naturally, however, and the attempts leave me puzzled. For example, Yan points to the presence of 2 restriction sites, ECORI and BST-EII, present at convenient locations in the novel coronavirus genome. Molecular biologists routinely use these enzymes to cut DNA and insert DNA into a plasmid or genome. ECORI is especially popular due to the reliability of the enzyme. Today, however, there are better methods to do the insertions. If one were working on a bioweapon and did use such a site, he would simply erase the evidence prior to releasing the virus. The presence or absence of such sites is meaningless. Unless it's I don't know. Similarly, why would someone leave 100% sequence identity in a bioweapon? So in other words, what this guy's saying is if they were trying to make it a bioweapon, then they would have hidden the fact that it's man-made better. A competent bioweapon engineer could erase any solid evidence from the genome. It would not take more than a week to do so. It is therefore baffling that anyone would attempt to even conjecture a natural origin when it could easily be made to look so. It certainly appears in the case that coronavirus E gene is solid evidence of unnatural origin. It was likely lifted from one of the strains, either ZC45 or ZXC21, and recombinantly placed or engineered into the novel coronavirus. But let's just accept this as highly suspicious, not proof. Pay attention as well to proximity. We must now combine this with the knowledge of the location of the outbreak epicenter. There are two labs, Wuhan Institute of Virology and Wuhan Center for Disease Control and Prevention, 8 miles and 300 yards from the officially claimed epicenter of the seafood market, respectively. It strains credulity that we have a rare natural recombination event that just so happened to occur between two labs that were performing recombinant experiments on coronaviruses. So, here's the thing. I'll get a little further. The virus is almost certainly created for research purposes. Why am I so confident of this? The U.S. National Institute of Allergy and Infectious Diseases provided funds to the lab in Wuhan through the EcoHealth Alliance to perform gain of function research. These types of experiments involve using a strain that is able to infect humans, systematically taking genes from it and inserting them into a strain that cannot infect humans. So why not just ask the Chinese scientists Jing Young Ma and Xing Shi, who headed research funded under the National Institute of Allergy and Infectious Disease Project? These scientists and others associated with the project are either missing or have not been made available for comment. Yet scientists with such relevant experience ought to be front and center of any response. 4 points indicate it was probably produced in and escaped from the lab in Wuhan. A highly suspicious gene sequence, the proximity of the epicenter, funding to create recombinant viruses, and missing scientists. So here's the thing. My NI is telling me this is correct, that it was in fact man-made and released accidentally. But because I'm a smart person and a good thinker, I checked the link on the alternative approach. This argues it's naturally occurring. But note what it does. Although sequence similarity values of 96 and 97% make it sound like the available bat viruses are very closely related, in reality this likely represents more than 20 years of sequence evolution, although the underlying molecular clock may tick at an uncertain rate if there was a strong adaptive evolution, blah, blah, blah. It is therefore almost a certainty that more sampling will identify additional bat viruses that are even closer relatives. A key issue is whether these viruses or those from any other animal species contain the key RBD mutations and the same fern-like cleavage site insertion as found in SARS-CoV-2. In other words, I bet we're gonna find this same thing in other strains of COVID, so that we have another explanation than it came from this gen bank. I bet we'll find this ZC45 and ZXC21 in some other virus, and that will disprove this notion. That's why it's natural, purportedly. Okay, so now, again, look at CNN's, look at, this fact check thing. What does it say? It says, scientists outside China have had a chance to study the coronavirus. They concluded it does not show signs of having been manufactured. Okay, who are these scientists? And did they address the thing that I just read from? Actual arguments, like real arguments? There is no evidence to suggest the virus was created in the Wuhan laboratory. On April 21st, the World Health Organization noted at a briefing that available evidence at the time on April 21st indicates coronavirus originated in animals and was not manipulated. That's April 21st, and this is October 29th. Okay? And then it says here, what's the date on this thing? Anyway, the Scripps research study that rejects the notion the virus is man-made. It says if someone were seeking to engineer a new coronavirus as a pathogen, they would have constructed it from a background of a virus known to cause illness. But the scientists found that SARS coronavirus 2 backbone differed substantially from those already known coronaviruses, or mostly resembled related viruses found in bats and pangolins. There's no notion here. There's no notation here of the fact that two of the elements of this are genetically identical and appear to be taken from a gene bank. No response to it. No reference to it. They're not actually. Let's talk about straw manning, right? Scientists from multiple countries overwhelmingly conclude nothing's addressing the actual argument. 2019 COVID is 96% identical at the whole genome level to a bat coronavirus. Okay. Was closely related to two bat-derived severe acute respiratory syndrome-like viruses. Researchers concluded the genome sequences of the coronavirus was closely related to. Okay, they didn't say was closely related to, they said identical. Identical to ones that are found in a gene bank. So, you'll note the avoidance of the actual arguments here. The straw manning and the refusal to address the fact that this would appear to be something of a smoking gun. If I take genetic sequences that are identical to ones that are available in a gene bank of some sort, that are used in virus manipulation, in research and such, it's a pretty good indication, apparently, that in fact, something's made by human beings. So, like, okay. So then, should we treat it differently, Eric? Well, sure. Is our response to COVID different? Well, not really. But I hate the fact that the media is pushing an agenda rather than going with the truth. It's pretty clear. That's pretty smoking gun. It's not absolutely definitive, but my NI is saying, ding dong, this is clearly man-made. I give credit to Soul, who argued with me and tried to convince me that it was man-made yesterday. And I looked into it more, and I hereby stand convinced it's a man-made virus, probably released accidentally.